Peace be upon you, ninth graders. In this video, we will study about enzymes, the agents of digestion. So prepare your materials, follow the instructions, and let's get started. At the end of this video, you will be able to determine the necessary conditions for the normal functioning of an enzyme, define enzyme, distinguish between digestive juice and enzyme, list the properties of an enzyme. In the previous video, we learned that salivary amylase is an enzyme that transforms starch into maltose. Is salivary amylase the only enzyme present in our digestive system? Does salivary amylase digest all kinds of food? Do enzymes need specific conditions to function? Well, all of these questions will be answered as we proceed with our video. In order to study the mode of action of digestive enzymes, we conducted the following experiment. So here in tube A we have cooked starch and fresh saliva, and in tube B we have albumin of an egg and fresh saliva. But what is albumin? Well, it is the white part of the egg, see? Now let's get to the description. Fresh saliva is put in two test tubes A and B. Cooked starch is added to tube A and albumin of an egg is added to tube B. The two test tubes are placed in a water bath at 37 degrees Celsius for 1 hour. Notice that we put saliva with starch and albumin. We previously know that saliva acts on starch, but does it act on albumin? Is saliva specific for starch? What is the substrate of saliva? Does saliva act on egg albumin? So our hypothesis would be saliva does not act on egg albumin, or you can say saliva acts on egg albumin. Before we proceed with our experiment, let's learn how to make a table of conditions. The conditions are variables like temperature, time, juice, substrate, enzyme that are relative to each test tube. You start by drawing a table with two entries. You put the tubes on a side and the conditions on the other side. So here for tube A, we have the substrate is the cooked starch, while for tube B, the substrate is the albumin. Juice is saliva for both. Temperature is 37 degrees Celsius for both, and the time is 1 hour for both. And don't forget to give a title for your table. Let's now indicate the variables. Remember, the controlled variables are the common variables in both test tubes. This would be the type of juice, which is saliva, the temperature, and the time needed. However, the manipulated variable is the variable that's different in both test tubes, and this would be the type of the substrate. As for the responding variable, it is the result that we are waiting for, which is the transformation of starch and albumin. After 1 hour, the Fehling test gives a brick red precipitate in tube A, indicating that the starch in tube A has transformed into a reducing sugar. However, Biuret test in tube B gives a violet color. Remember, a violet color with Biuret test indicates the presence of proteins. But tube B previously contained protein. Remember, it contained albumin. So we can deduce that the albumin in tube B is still present. It is not digested. After 1 hour, there is formation of a brick red precipitate with Fehling test in tube A that contains cooked starch and fresh saliva. This means that tube A contains a reducing sugar. However, Biuret test gives a violet color in tube B that contains egg albumin and fresh saliva. This means that tube B contains protein. Derive a conclusion concerning the tube where digestion took place. Saliva digested starch into reducing sugar in tube A, but didn't digest protein in tube B. So the first enzymatic property, enzymes are specific. Each enzyme acts on a specific substrate. So the salivary amylase in saliva is specific for starch and not for proteins. This is all for today. We will continue the objectives of this video in the next session. So, watch the video again, and if you have any questions, you can contact your teacher. Study well and keep safe.